kind of unbelievably naive behavior. I don't know what to tell you. Good evening, universe. Good evening, Pittsburgh. This is JC on a bike. My name is JC, and this is Journal Club on a bike. On my way home from the University of Pittsburgh again on this Wednesday evening. And I'm going to do one of my general journal clubs again, this time on a science paper, which seems to have made a pretty interesting discovery about a very important neuropsychiatric disorder or condition. The largest cause of genetic mental disability is apparently trisomy 21 or Down syndrome. And Down syndrome, as belied by the name, nice. As belied by the name, is a genetic disorder where you have an extra chromosome 21. And so the mental deficits and learning difficulties and all of the mental issues associated with Down syndrome have been historically assumed to be directly related to the genetic aberration that is that third chromosome, that extra chromosome, the trisomy condition. But interesting anecdotal evidence has for a long time pointed elsewhere. Perhaps the best of this anecdotal evidence is the idea that the genes that are misregulated in Down syndrome are not only genes that are on chromosome 21, already suggesting something else changes in this condition that doesn't have direct relationship to this extra genetic material. So these authors are interested in the idea that the integrated stress response, which is a molecular circuit in all cells and certainly in the brain, a molecular circuit which regulates the homeostatic transcription of proteins. In case you are unaware of this idea, the brain and the body in general are required to constantly manufacture proteins because proteins, through their use, can become destroyed, broken, in need of repair. And this repair occurs primarily by replacement. AMPA receptors, NMDA receptors, enzymes, transport proteins, scaffold proteins. Any protein that goes through some kind of conformational change or provides some structural or enzymatic backbone is going to necessarily be exposed to chemical reactions that will eventually cause it to degrade and be necessary to be replaced. This protein homeostasis is a well-known and pretty well understood idea, in the sense that if you don't have protein synthesis, then all kinds of things go wrong in the brain. And so these researchers from Baylor University and from the University of California, San Francisco, in this paper put forward the hypothesis that the integrated stress response, this molecular cascade, molecular circuit, which regulates protein homeostasis, the integrated stress response is activated in an abnormal way in the Down syndrome condition, which results in poor maintenance of proteins in critical regions of the brain, including the hippocampus, which ultimately result in the deficits in these animals. So their idea then, and so first they look to see if any regulatory elements of the integrated stress response are activated in Down syndrome patients or in a mouse model of Down syndrome. And what they see is that key proteins and key enzymes that are involved in this integrated stress response are indeed activated, overactive, or active in consequential ways in the Down syndrome model and also in human Down syndrome patients. So that's pretty spectacular. And even more so, they show that even cultured cells from the humans or cultured cells from the mouse model will also show a similar integrated stress response reaction, which is upregulated from the baseline. And so this basic finding then gets explored in terms of causality and necessity in this paper. So the way they're measuring this response is by the ratio of a phosphorylated regulatory element to its baseline state, I think. And the more phosphorylated this element is, thank you, then the more integrated stress response is activated. So in figure 2, they just look at that. And what they do in figure 2, I think, is that they cross this mouse model with a mouse model that has the primary activating enzyme, the phosphorylating enzyme for this cofactor, EIF2. They remove this phosphorylating enzyme by interbreeding the mouse model with this knockout mouse and selectively ablate this phosphorylating enzyme and mostly rescue the phenotypes of the mouse model. So without the enzyme, it starts to normalize this mouse model that recapitulates a lot of what Down syndrome does. So in figure 3, they go a little bit farther, and besides showing sorry, I want to get through here first. Yeah, sorry. So in this second figure, they're already showing a lot of the memory deficits being rescued, including context fear conditioning and including novel object test and a field potential experiment in slices. And all of these reveal the same thing, that more or less knocking out this enzyme which phosphorylates which starts the cascade more or less of the integrated stress response and downregulates proteome homeostasis. This enzyme, if they knock it out in this mouse model of Down syndrome, then a lot of the phenotype of this mouse model is normalized. It's not completely normalized, but it goes in the direction of normal. Now, if you couple that with the fact that they can also look at all the proteins that are expressed in such a mouse and try to compare them to the proteins expressed in the mouse model alone without the extra knockout of that enzyme, they can see that when they knock out the enzyme, that the maintenance of the proteome kind of returns to normal, and a lot of the levels of the proteins that are expected return to normal. That's figure 3. So that's a pretty extraordinary correlate to figure 2, where the physiology is also partially redeemed, to show that all of these proteins are not as downregulated as they were in the original model is a spectacular result. And so already the paper is devastating in the sense of how clear the results actually are. It's an extraordinary set of figures. Unfortunately, he didn't know he could turn there. So in figure 4, they're going to do the same thing, but now they're going to try and dysregulate the regulatory element EIF2 by interbreeding it with another mouse wherein EIF2, the protein itself, only half of the proteins that are manufactured are available for phosphorylation. So by this manipulation, they essentially reduce the phosphorylated EIF2 by roughly half. And that reduction leads to almost complete, it's essentially statistically a complete correction of the deficits they measure in context fear conditioning and in novel object test and in the field potentials, TP. So they've done 2 different ways of using genetic tools to manipulate the integrated stress response, and shown that in a mouse model of Down syndrome, they can indeed reverse these things. And then in the same figure, I think this is still figure 4, they use some pharmacology to do the same thing and show that the pharmacology has a similar effect. And so in the final figure, what they look at is inhibitory tone and spontaneous inhibitory events as a way of seeing whether there has been a kind of circuit change. One of the things that's known about this mouse model is that inhibitory tone is increased, I think. It's decreased. Shoot, no, I don't remember. I don't remember, I gotta blow my nose anyway. Excuse me for a second. Yeah, so in this mouse model, there's an enhanced inhibitory tone. And this enhanced inhibitory tone is essentially normalized again by the manipulations from figure 4. And so all of these figures point to the idea that something changes in terms of protein homeostasis in response to the extra chromosome in Down syndrome, and that leads to the general effects of Down syndrome. This is a really interesting observation if it holds true for human Down syndrome, that the main effect of this extra chromosome is to change the way that the proteome is expressed, rather than, overexpressing particular genes that are on this chromosome, which I think a lot of naive and talented geneticists have thought for a long time, that extra genes are what's the problem, right? But it's actually the system's reaction to these extra genes that's the problem. It's not the extra genes themselves, but the system's reaction to them. And this paper really seems to suggest that this Dr. Walter guy has got the right idea. Apparently it was his idea. That an integrated stress response causing a lack of protein homeostasis can lead to all or many of the deficits seen in Down syndrome. And none of them are directly necessarily related to any genes on chromosome 21 per se. So that's a science paper, and that's wicked bad. This is the kind of work everybody dreams of being able to do, and the kind of he had a Trump 2020 sticker on his car already. You gotta wonder who those people are. They're just sad, angry people, I think. Not very many loving, caring, gentle people whom you'd want to be your neighbors are Trump voters. I mean, they're really not. Let's just be honest. So this was a paper from Baylor and from the University of California, San Francisco. And I think it's a really good paper.